Welcome to our virtual lab tour for the T-cell Modulation Group. In this video, we're going to tell you about our research, our interests, why we love science, and what we do on a day-to-day basis. As you know, we're based at Cardiff University within the School of Medicine, and you can find us here in the Henry Wellcome building. This video gives you a quick look around our multidisciplinary labs. Our primary research focus is the T-cell. In our tissue culture labs, we look at T-cell biology. In biochemistry, we study genetics and proteins, and we work on structural biology in our crystallography labs. We are a multicultural research group with a diverse skill set and knowledge base. The team is led by Professor Andrew Sewell and includes researchers from different scientific backgrounds at all stages of their careers. Before we tell you about our work, we're going to set the scene with a brief introduction to the immune system and what exactly T-cells are. You will have heard lots about the immune system in the news over the last 12 months, so you know that it's our natural defense system. We study T-cells and the important roles these cells play in fighting the diseases caused by bacteria, cancers, autoimmune diseases such as type 1 diabetes, and of course viral infections like coronavirus. The immune system is divided into two arms, the fast-acting innate system, which is called into action as soon as a threat is detected, and the slower adaptive response that provides long-term immunity. T-cells are like an army on patrol waiting to be called into action by a signal from the innate immune system. In the video, you can see the T-cells arrive and use their own version of X-ray vision to look for a threat. Each T-cell has a protein on the surface, the T-cell receptor, which they use for seeing inside cells. The T-cells in the video are killer T-cells. Their response is to kill the cells they've discovered to prevent the further spread of disease. These cells go red and break apart when they die. Last year, we published some work that caused an international stir. First, it was reported by the BBC, and then it went global. Inboxes crashed, social media went crazy, and film crews took over the labs. Let's hear from the boss about why this work caused such a media frenzy.
So we found a new type of white blood cell that has the capacity to kill most cancers from all people.
This is the story of how our team found that T-cell. We named it after the scientist who found it, Mike Crowther. The T-cell is called MC7G5, although that's a bit of a mouthful, so we're going to call it 7G5 for the rest of this video. We used a multi-pronged approach to find this T-cell, calling on many different techniques and using the wide skill set of the team. The first step in our research is to extract immune cells from blood donated to the Welsh Blood Service. The plasma part of the donated blood was separated and used to treat patients, then we received the remaining part containing all the cells of the immune system. This video shows the process of separating and extracting the white blood cells from other parts of the blood. You can see the tubes of blood changing colour as the samples are diluted in media and spun at high speeds in a centrifuge to separate the blood cells by density. The red blood cells collect at the bottom, while the white blood cells can be removed from the middle of the tube. These white blood cells were cultured along with some cancer cells in a T-cell friendly environment. With some TLC over the next few weeks, the T-cells in these cultures survived because they recognized the cancer cells which stimulated their growth and expansion. This is similar to the way that T-cells would survive and expand within the body in the presence of a cancer. We then used some clever lab tricks with fluorescent antibodies. The antibodies acted like a claw, grabbing those killer T-cells that had survived and proliferated in the presence of cancer. Amongst these cells, we found 7G5. To assess the functionality of 7G5 T-cells, they were tested against a wide panel of cancer cells from different organs and tissues. It took months and months running the experiments to find the red trail that shows up when a T-cell kills a cancer cell, with each one of these experiments repeated for every different cancer cell line. 7G5 passed the test, killing cells from the most common cancers, lung, breast, colon, and prostate, as well as cancers with a high mortality rate like pancreatic cancer. Next, we had to show that 7G5 would only kill cancers and not the healthy cells from the same parts of the body. So we did all those experiments over again, this time with the healthy cells, and showed that 7G5 doesn't kill healthy cells. And to conclusively prove beyond any doubt that this T-cell would kill cancer but not give any negative effects on healthy cells, we used a live experimental model. For this, we used a genetically engineered mouse. These mice are sedated while a detector looks for signal from their cancer cells. The mice on the right received some of the 7G5 T-cells, and those T-cells killed the tumour without causing any unwanted side effects to the mouse. We had spent a long time finding this T-cell and finding out what it could do, so we didn't stop there. We went on to find out how it worked. An emerging technology helped us to discover the mechanism of action of 7G5. CRISPR-Cas9 uses genetic scissors that specifically cut part of DNA that encodes proteins, which in turn interrupts the production of the protein and creates a genetically engineered cell. We can label the cells with a barcode that allows us to trace it. This huge bank of genetically engineered barcoded cancer cells was systematically tested, like you can see in the video, and we found the cancer cells which could now survive 7G5. We read the barcode of the surviving cancer cells and found the 7G5 target protein. We had found 7G5, we knew it was great at killing cancer, and now we knew its target. Next, we wanted to further get into how this T-cell works by studying it at a molecular level. We used more genetically modified cells, this time laboratory strains of E. coli, which aren't dangerous and had been engineered to make proteins. We designed a DNA message to tell the E. coli to make the 7G5 T-cell receptor protein, and some other E. coli to make the 7G5 target protein. These bacteria made a lot of protein, which we then refined and purified until we had enough T-cell receptor and target protein to make protein crystals. Why did we make crystals from these proteins? So we could make a picture like the one in this video. It shows the shape of every molecule in the protein and how the molecules meet. We can see which parts of the protein will touch and start the process of 7G5 killing a cancer cell. We grow crystals of the T-cell receptor and the T-cell receptor bound to its target. The molecules in the protein crystals diffract light, and we can use this property to our advantage. Using X-ray light at the diamond light source synchrotron, we measure the scatter patterns made by the diffracted light, and you can see one of these here on the right. The scatter pattern shows the shape of all the molecules in the protein. It sounds incredibly technical, and it is, but it's also fully automated. We can sit at our desks here in Cardiff and remotely operate the robot in Oxfordshire. We're very lucky to have such amazing facilities like Diamond Light Source and their scientists available to us. What comes next? We have the T-cell, we know what cancer it kills, we know its target, and we know how the T-cell receptor interacts with the target. We really hope that 7G5 could be of therapeutic use. This is still a long way off, but let's look at how it could be used. 7G5 is a good candidate for a type of immunotherapy called adoptive cell transfer. The cartoon on the left shows how a specially generated virus is used to send a DNA message to the patient's own T-cells. The patient's T-cells would start making the 7G5 T-cell receptor, beefing them up, ready to defeat the cancer. The patient's cells would be multiplied in a lab before being given back and outnumbering the cancer, making it impossible for the cancer to survive. With our new partners, Inara Bio, we hope 7G5 will make it to a small trial before building it up to include more and more patients. Eventually, we hope that 7G5 would pass all the tests and make it to become a new cancer drug. So, to recap our work with the 7G5 T-cell, we found that it attacks and kills many different types of cancer cell, but it does not kill healthy cells, and this is what makes it a good candidate for clinical trials. We didn't do this alone, and we could never have achieved our objectives without our amazing collaborators. This map shows the locations of some of them. You can also see a selection of past team members who've moved on to work across the globe in locations as far as New York and Australia. A career in science creates the possibility to work in many different locations. Just pick the one you like the most. Thank you for watching. We hope you enjoyed that whistle-stop tour of the processes we use to find clinically interesting T-cells. This is our team and our funders. If you want to find out more about us and our work, then please visit our website at www.tcells.org or follow us on Twitter.